Okay, so today we're going to discuss all about the anaerobes. Okay, so we have here the different types of the anaerobes. So we can have here your aerotolerant anaerobes. Again, your aerotolerant anaerobes are anaerobes or those bacteria wherein they can survive in the presence of the oxygen because diba, anaerobe, those are the bacteria wherein they survive only in the absence of the oxygen. But in the case of your aerotolerant anaerobe, those are the anaerobes wherein they can survive in the absence of the oxygen or in the presence of the oxygen. Only that, in the presence of the oxygen, they are not performing metabolic activities. Your facultative anaerobe, on the other hand, so those are also anaerobes wherein they can survive in the presence of the in the absence of the oxygen, of course, being an anaerobe, but they can also survive in the presence of the oxygen. Only that it differs with your aerotolerant anaerobe because your facultative anaerobe can perform metabolic activity even in the presence of the oxygen. Of course, they can perform the metabolic activity in the absence of the oxygen. But in the case of your facultative anaerobe, they can still perform metabolic activity even in the absence of the oxygen. But unlike your aerotolerant anaerobe, they can only perform metabolic activity in the absence of the oxygen. But in the presence of the oxygen, they cannot perform metabolic activity, but they can survive. Okay, we have here the term obligate or strict anaerobe. So again, pag obligate or strict anaerobe, so basically they will die in the presence of the oxygen because oxygen is toxic to this type of your anaerobe. Okay, paano nagiging toxic ang oxygen sa kanila? Okay, normally for our bacteria, okay, so for other bacteria, we have here your elemental oxygen it's being converted to your superoxide anion. Okay, then eventually your superoxide anion here try to uh, reduce here to be converted to your hydrogen peroxide. Okay, so once they're converted to your hydrogen peroxide, okay, so in the case of your okay, either that's the usual na okay na production of your oxygen or that will be the usual na metabolic process with your oxygen where your elemental oxygen is converted to your superoxide anion and eventually converted to your hydrogen peroxide. And superoxide anion and hydrogen peroxide are your superoxide radicals. Those are your free radicals which are toxic, very toxic. However, in the case of your other bacteria, they possess the enzymes wherein they can tolerate the presence of this even toxic na mga byproducts of your elemental oxygen because they have an enzyme. Okay, so we have here your superoxide anion is eventually converted to your hydrogen peroxide if they have okay your enzyme superoxide dismutase. Okay, so even if they are producing superoxide anion for some other bacteria, they can convert that one to your hydrogen peroxide because they have the enzyme superoxide dismutase. And eventually, their hydrogen peroxide formed here could be converted further to other products like water and oxygen by the enzyme catalase. And this production of your, although you have your oxygen here, but it's less toxic compared to your elemental oxygen compared to your superoxide anion. If that is your, usually this one occurs in our bacteria which are aerobic. So they can survive in the presence of the oxygen. They can tolerate the presence of the oxygen even if that one is toxic na oxygen because they can convert your oxygen to a byproduct which is less toxic. Because again, they have the enzyme this superoxide dismutase and catalase. But in the case of your case of your strict anaerobe or your obligate anaerobe, they don't have yet the enzyme. So wala silang superoxide dismutase. So once you have the presence of elemental oxygen, your elemental oxygen will be converted to your superoxide anions and hydrogen peroxide. And again, those are toxic. And therefore, mamamatay sila. But for some other bacteria like your aerobes, they can have here, although in the presence of oxygen, they can still tolerate that one because they can convert your elemental oxygen to other byproducts which are less toxic. Because again, they have the enzyme, superoxide dismutase and catalase, which are, again, absent among your strict or your obligate anaerobes. And therefore, once oxygen is present, your strict or your um, your obligate anaerobes will die in the presence of the oxygen. Okay, we have here the distribution of our anaerobes. So anaerobes can be found here in the environment outside our body. We call that ones are exogenous. Most likely, we get infected by contaminated food or even soil, for example here, or objects which has been contaminated by your anaerobes. Okay, for the exogenous distribution of your anaerobes, most likely they are found in mga Clostridium species natin. So your Clostridium are basically those are with the spores. Okay, so you get infected by exogenous here by allowing the spores to enter your body through contaminated objects, like in the case of your tetanus, or even contaminated food in the case of your perfringens, or even your botulinum. Okay, so again, they're found in the environment, they get entry to your body, and eventually they try to cause infection. Sample for that, we have your contamination of your wound by the toxin or by the spores coming from a source. Okay, like contaminated object here, like your Clostridium tetani that try to cause your tetanus, or even your Clostridium perfringens through food which has this contaminated with a preformed toxin. If you try to ingest that one, you get infected. Another one, ingestion of the preformed toxin in vegetable or meat. Ay, wait lang. Clostridium perfringens is by also an object contaminated by the preformed toxin. Or even the food which has been eaten by the patients containing here the preformed toxin, you get also infected. Like in the case of your Clostridium botulinum, or even the food poisoning related to your Clostridium perfringens. Or also colonization of the GI tract by the toxin producing organisms, like in the case of your Clostridium, your botulinum in the infant botulism by ingestion of the honey contaminated by your preformed toxin of Clostridium botulinum. On the other hand, we could also have endogenous. Pag sinabi endogenous, again, these are found inside our body. And basically, they're found as a normal microbial flora in a different part of your body. But remember that our normal microbial flora, they can get you infection if the normal body site has been damaged, where this organism can gain entry to your other body sites which are sterile. So, meaning to say, pag pumunta sila sa ibang part which is not usually inhabited by the bacteria, you call it a sterile site, then they cause here infection. So, example for that na sterile body site natin, so you can have your bloodstream. So, bloodstream, wala tayong bacteria dapat in our bloodstream. Or even, they can gain access to your lungs or even your brain. So, wala talaga dapat bacteria, Anton. So, normally, okay, these are the normal anatomic site where you can find this mga anaerobes natin. But then again, they can cause infection if that tissues has been damaged, maybe because of the trauma, because of the surgical operation. Okay, example, we have the skin. So our skin is normally inhabited by our Propionibacterium acnes. So again, they can tolerate that one area because they have, they have the enzyme lipase, diba? Wherein they can digest the lipid component of the skin. Okay, and therefore, they can inhabit that area. Our respiratory tract is normally inhabited by the following. We have your Prevotella, Porphyromonas, and we have also your Fusobacterium. Gastrointestinal tract is being inhabited here by Bacteroides fragilis and your Clostridium difficile. And your genitourinary tract is normally inhabited by our Bacteroides, Prevotella, and even your Fusobacterium. Okay. Okay, then we have here the specimen quality. So, again, it's very important that we need to make sure that the collected specimen would not be exposed to the oxygen environment. Okay, so most likely, our anaerobes are being taken from the moist and dry, moist area in our body. And most likely because meron siyang moist area in our body. And therefore, you need to keep that one in the environment free of the oxygen because once they are exposed to oxygen, they die. Okay, so the best specimen collection method here is to collect the specimen directly from the area where there is an infection. Okay, so to prevent here, most likely by contamination with the other surfaces. So therefore, ang best sample collection natin dito is not by the swab, but rather by aspiration para directly to the it would have here collected the specimen directly to the tissues where it will try to cause infection. Or pwede din siya ang tissue biopsy natin. Okay, but then again, your swab is not a suitable specimen for collection process because again, the swab might be contaminated. So pag nag-swab tayo, di ba, like in the skin surface, okay, it might not be really the one na nagkakause infection, might be contaminated with the other microbial biota found in our skin. So therefore, we need to really minimize that one. Okay, we have here your unsuitable specimen. So we have your swab, voided or catheterized specimen, even your fecal sample, provided it's not used here for the following. So pwede ang fecal sample natin pag, of course, food poisoning, diarrhea ang patient natin. We have also here cough expectorated na sputum, bronchial washing. Okay, again, they are not suitable for the collection process our specimen because again, it might be containing here the other na samples. It might mix up with actual na organism which try to cause infection. That's why we advise that you need to collect it directly to the area by aspiration mo para direct siya. Na yun talaga makukuha natin. Or by your tissue biopsy. Again, your fecal sample could be used as your sample for your anaerobic culture for the following infection caused here by our anaerobes which are causing our food poisoning or enteritis. For the first one, we have here food poisoning related to the caused by Clostridium perfringens. Okay, another one is the food poisoning here brought about by Clostridium botulinum, the food in your food botulinum. We have also here your pseudomembranous enterocolitis caused here by Clostridium difficile. And we have also your neutropenic enterocolitis caused here by Clostridium septicum. Again, those are all related to our gastrointestinal tract infection. So most likely the patient try to manifest diarrhea with this one. So you don't have the choice but of course to collect your stool sample for that. But ideally dapat in this stool sample ang ating specimen. Okay, then we have also here your specimen transport. Okay, so when you're collecting the specimen, make sure that one you maintain that one in culture or in transport media that is reduced or with absence of your oxygen. So the first one, you could have here your rubber stopper collection vials. This is a rubber stopper. Okay, so that is ideal here for collection of specimens which are your body fluids or even your pus. So medyo liquid na specimen. Example for that, we have here port equal by the BD Diagnostic Company. You could also have your PRAS or the pre-reduced and anaerobically sterilized na tube. So pre-reduce means to say try to reduce already the oxygen content. So walang oxygen content on that. So example for that, we have here your rubber stopper vial system. So meron siyang rubber stopper. Okay, that will prevent, of course, the introduction of the oxygen para walang oxygen because again, your oxygen is toxic to our anaerobes. Then you have also here the agar system. The agar system also provides you with, of course, that would allow here the growth of your organisms. Okay, so this is agar. Okay, then you could also have your oxygen-free transport tubes or the vial. This is ideal here for the swab na specimen. For example, for that, you can have your vacutainer, okay, by the BD Diagnostic Company, but also the press. Again, you can insert your swab on that, but most likely, di ba, okay, swab is not an ideal specimen, but if you don't have the choice but to collect the specimen, the swab, you can transport that one, put that one here in your oxygen-free transport tubes or vials. Okay, so another one, we have your anaerobic pouch. Parang pouch lang siya, tapos may seal mo lang siya dito. So this one can accommodate at least 2 na mga plates. So once you try to put on your plates inside your pouch, since this one is clear naman ang ating pouch, you try to seal that one and then try to activate your oxygen reduction system. So para walang oxygen na papasok or you're able to remove the oxygen on that. So example for that, we have your gas pack pouch by the BD Diagnostic. Again, this is a deal here for our tissue specimen. Okay, your pouch, pwede siya magiging anaerobic jar or even anaerobic na chamber. So this could serve already as its area where you try to incubate. Okay, then only that, ilalagay mo lang siya dito, then try to incubate at 135 But then again, because of the pouch, it try to provide an oxygen-free environment. So maganda nito ay, of course, clear siya so you can observe for the morphology of your bacteria growing the plates without taking their plates out of your pouch. Kasi pag tinanggal natin, of course, the environment, may oxygen tayo, so baka mamamatay ang ating specimen, ang ating bacteria. Okay, we have here the content of our transport media. So number 1, reducing agents. Reducing agent, try to reduce the oxygen in the environment. Okay, so example for that, we have your sodium thioglycolate, your cysteine, and your that's your DTT, dithiotritol. Another content would be your indicator system. So we have your redox indicator. So the indicator system will try to be the one as our a marker. Actually, that will help us to check if your entire environment has been contaminated with an oxygen or wala. Sample for that indicator system, we have your resasurine. Another one, you could have also your methylene blue, wherein they try to change the color. Resasurine, nag-change ang color niya from colorless to pink. Pag wala siyang oxygen na environment mo, resasurine will be colorless. Pero pag na-contaminate yan ng oxygen, so magiging pink na siya. So of course, pag na-contaminate ng oxygen, mamamatay na sila. Methylene blue, on the other hand, would change the color here from your white pag walang oxygen. And blue, magiging blue siya pag may oxygen. So that will help us then to know if the environment where you try to put your culture media, okay, or your cultured or your isolated specimen or organisms would have here the oxygen present on that or wala. So it could help us then. Because remember, dapat wala tayo, we should maintain the environment here with your chamber or incubator and all. We try to put your specimen na wala dapat siyang oxygen. Okay, then we have here the processing of our clinical specimen. So once you have already culture media, growth of your bacteria, you can have here the colony characteristics for identification of the specific na mga or have the presumptive identification of your specific na anaerobes. Because each of them would have here some morphology characteristics unique to each of your anaerobes. The first one, you could have your foul odor. So in the culture media, if you try to open that one, so you can have your foul odor produced here by the following. Number 1, you could have here your Clostridium difficile, you could also your Fusobacterium, you could also have your Porphyromonas. On the other hand, others will try to produce here brick red na fluorescence, one subjected here to the long UV lights. So that's in the case of your Prevotella, Porphyromonas, or even your Veillonella, which is the gram-negative gram-negative cocci na anaerobes also try to produce here brick red na fluorescence. On the other hand, you can also examine the tissue here. So if the tissue submitted, for example, is black, or maitim ang tissue na kinolect ng galing from your patient, so it might signify infection related to your Prevotella and even your Porphyromonas. Another one, sulfur granules. So we are considering your actinomycetes, just like our discussion in your nocardia, diba? So that one would have also sulfur granules because may pus siya na yellow-orange na pus that would resemble our sulfur granules. Or even your Propionibacterium acnes. Also try to produce here your sulfur granules. Then we have also here the microscopic examination. So you can perform your gram stain, and the results of the morphology under the gram stain here would also help you to really provide you with some information that would help you identify and confirm your test result. Number one, to determine the polymicrobial infection. What do you mean by that? So if you happen to identify under the microscope with your gram stain smear na ay may mga gram-positive cocci, may nakita kang bacilli, may nakitang gram-negative. So mean to say there are polymicrobial. Mean to say when speak about polymicrobial, madami. Madami ang anaerobes which are the one causing the infection to the patient. So pag isa lang na klase like gram-negative bacilli lang, so isang klase lang siya. Hindi siya polymicrobial. The second one, to guide for the means of selection. And the third one, to provide presumptive identification. Like for example, if you happen to identify the presence of your large gram-positive bacilli na may spores. So we are considering the presence of our Clostridium. So pag Clostridium ito, you need to inoculate that one to another culture media. So kailangan meron kang culture media with your egg yolk agar in order for you to identify the test result related to your lecithinase test. And we have also here the lipase na test. Again, lecithinase test and the lipase test could only be visualized in your egg yolk agar. And that's very important. It's needed when you're having that Clostridium na isolate. Another one, if you happen to identify gram-negative, tapered, fusiform. Okay, so pointed ends. So one end is pointed, the other one is tapered. So we are considering the presence of your Fusobacterium nucleatum. But if that one is bizarre, pleomorphic, iba-ibang shape, morphology, then that's your pero gram-negative din siya. So it might be related to your Fusobacterium mortiferum and necrophorum. Another one, if you happen to identify tiny or small na gram-negative cocci, so that might be related to your Veillonella. So for the gram-negative naman, but they are Coccobacilli, that includes your Bacteroides, Prevotella, Porphyromonas. Another one, the gram stain result can reveal also here the presence of the leukocytes. Of course, the leukocyte will always be related to your inflammatory reactions or even to your pus-producing na mga infections. But then again, if you happen not to identify the presence of the leukocytes, do not rule out na wala siyang inflammatory process, because remember that there are some bacteria wherein they are producing a virulence factor in the form of your leukocidin. So leukocidin try to lyse leukocyte, diba? Leukocidin try to lyse is a hemolysin which try to lyse your leukocyte or WBC. So pag meron siyang leukocidin, do not expect na may makikita ka pa na WBC or pastas within your smear. Kasi diba, again, they tend to destroy, the leukocidin tend to destroy here the leukocytes. Another one, pag sobrang dami ng squamous epithelial tissues found in the specimen, it is a sign of contamination. Meaning to say the patient, or the medtech did not collect the sample correctly. So, baka swab siya instead of aspiration. Ganun. Okay, then we have also here our culture media and our mga plated na culture media that also helps us then to identify our anaerobes. So, most likely, when we tend to inoculate our specimen, you need to inoculate that one in your non-selective na mga blood agar. Sample for that, we have your Brucella blood agar. Okay, another one, you could also have your phenylethyl alcohol and your CNA, cholesterol, nalidixic acid. This is for the gram-positive. Diba? It will not allow the growth of your gram-negative for both your phenylethyl alcohol and your cholesterol, nalidixic acid agar. Okay, another culture media here, we have your anaerobic broth. So first one, we have your thioglycolate. Thioglycolate will allow the identification of your different types of the anaerobes. Your strict aerobe here, your strict aerobe, tama ba? Aerobe. So may air. So basically, they grow here on the upper surface kasi dito may oxygen sa taas. Your aerotolerant, your facultative anaerobe on the other hand, so this one grow here. Basically, okay, this one, here on above your, okay, above your, slightly above your surface. Your aerotolerant anaerobe, on the other hand, they grow throughout. And then your strict anaerobe, so dapat walang oxygen. So dito sila sa bottom ng tube natin. Again, pag strict aerobe nasa taas, pag facultative anaerobe, so medyo at upper surface and slightly below that one, your aerotolerant anaerobe were able to grow throughout your thioglycolate medium or broth, and whereas your strict anaerobe were able to grow here at the bottom of your tube kasi walang oxygen dun sa baba. You could also have your other na broth medium. We have also your cooked meat. Again, this is non-selective. So, ginagamit siya for the determination of the metabolic byproducts of your different anaerobes using your gas liquid chromatography. Another one, you could also have your peptone, yeast extract, glucose broth. Okay, that one would have also non-selective for identification also of your metabolic byproducts by our gas liquid chromatography. So, pareho lang sila. Another one, you could also have your kanamycin, vancomycin, lake blood agar. This is a type of the culture media wherein it contains 3 antibiotics: kanamycin, vancomycin, and your colistin. So this will help support the growth of your Bacteroides, Prevotella, and your Fusobacterium. Your Porphyromonas will not grow. And your kanamycin, vancomycin, lake blood agar. Your BBE or Bacteroides biosculin agar is for identification of its ability to grow in the presence of your 20% na bile. So, dapat ano siya, bile tolerant. And then, try to hydrolyze your esculin. So, this will identify your Bacteroides fragilis being able to be tolerant to your bile na 20%. So, pag tolerant siya sa bile na 20%, it grow. It will grow here in your biosculin, Bacteroides biosculin agar, and try to produce here black na colony because it tried to hydrolyze your esculin. Once it tried to hydrolyze your esculin, it result to your blackening of your colony, and that identifies your Bacteroides fragilis. Another one, we have your CCFA or your cefoxitin, cycloserine, cefoxitin fructose agar. This is a selective na culture media for our Clostridium difficile. This will identify Clostridium difficile here having large and try to produce here bright yellow fluorescence. If you try to subject that one to your UV light with your neutral red as a color as our indicator. Then we have your egg yolk agar. So this one is for identification of the lecithinase test and for also for our lipase test. For the lecithinase test, this is the basic reaction. Lecithin found in the egg yolk, if your bacteria contains the enzyme lecithinase, will convert your lecithin to your diglyceride. Positive result will be opaque zone around the colony. Pero hindi siya sa colony mismo nagkakaroon ng opaque zone, but in your culture. Okay, on the culture media, in the agar. Hindi doon siya sa surface mismo ng colony natin. Nagkakaroon ng opaque zone. To test for the lipase, on the other hand, so ito ang ating chemical reaction. So, our substrate would be your triglyceride, triacylglycerol or triglyceride, which is acted upon by our lipase, converting that one to your glycerol and your free fatty acid. Positive result would be iridescence or sheen or water in the gasoline. Para siya, di ba, parang, matawag dito? Di ba your gasoline in the water, di ba nakita mo yung sa taas? Okay, para siyang yan, iridescent. I think this one is the best description, iridescent or sheen. Or you call that one as your mother of pearls. So, this one naman, ang reaction na ito ay nasa surface mismo ng culture media natin. Ay, nasa surface mismo ng ating colony, not on the medium or not on the agar. Okay, so result here, so our Clostridium perfringens will only give positive result for the lecithinase but not on the lipase. For Bacteroides fragilis, it will give all the negative result. And for your Fusobacterium necrophorum, this will give a positive result with the lipase but not with your lecithinase. Okay, then we have also here your anaerobic incubation. So, saan natin incubate ang ating culture media which has been inoculated or not yet inoculated by our specimen for recovery of our anaerobes. Okay, the first one, we have your anaerobic chamber. So, anaerobic chamber could either be a glove or gloveless. Kung sabing gloves, ito yung chamber na close lahat. Tapos, pag may sinabing gloves, glove box, so may gloves na siya, ipapasok mo lang kamay mo doon sa gloves. So, and then you try to manipulate your processing, inoculation of the specimen within the chamber. Pag gloveless, so, ikaw ang sarili mong gloves. Okay, pero medyo mas fitted dito na area lang para pagpasok mo ng kamay mo, walang oxygen pa din na mapapasok or ma-introduce natin. So, the serve here is our storage or even inoculation of our culture media. So, for the growth of your culture media. So, like, if you have already inoculated your specimen in your plate, lalagay mo siya doon sa ating anaerobic chamber. Okay, and try to incubate usually at 35 or 37 at 24 to 48 hours and allow it to grow on that. So the content of our anaerobic inoculation chambers, or even all here, not only the chamber, it also contains the following. So number one, the gas requirements. So 85 to 90% na nitrogen, 5% na hydrogen, and 5 to 10% na carbon dioxide. Walang oxygen dito dapat. Another one is also contains catalyst ang ating chamber. Catalyst here, try to remove the residual oxygen that might be produces here pag nagkaroon ng water or even if some the bacteria previously has already or even the one you are inoculating is already producing metabolic products, we try to become here your water or the oxygen na magiging source ng oxygen natin. So, the catalyst within your chamber, try to eventually remove that residual oxygen para hindi siya mag-contaminate, mag-prevail or within that, pasok sa ating chambers. Okay, so ginagamit na ng catalyst, you could have your palladium-coated alumina pellet, or you could also have your, okay, that one. Usually, palladium-coated alumina pellet. Another one is being also provided by the desiccant to absorb the water. Again, it's also for the removal of the residual na oxygen. Desiccant usually used here would be your silica. Silica here nag-change ang color pero hindi siya ganun ka-advisable. But if you have the choice, you can go for this one. Solid state dehumidifier. Although, kaso mahal lang to. Medyo mahal ito. And then, of course, you could have your redox indicator that would try to be our basis pag nagkaroon ng oxygen contamination because this indicator try to change the color. So, the saccharine from colorless magiging pink and methylene blue magiging blue siya pag na-contaminate ang ating entire chamber, ang ating holding area or our inoculation area or chamber with the oxygen. Then you could also have here, so medyo mahal ang ating anaerobic chamber. So you can also have, so instead of the chamber, you can have your jar and bags na lang. So this jar and bags here are eventually provides an anaerobic environment provided here by the hydrogen and the carbon dioxide generator, envelope or by anaerobic or by evacuation replacement. Okay, so example for that, we have your brewer jar or gas pack system. Then we have also here the holding jars. So holding jars, it's the temporary holding area. So like, anong pilalagay mo dun sa holding jars? Itong mga plate na hindi pa na-inoculate. So itong mga plate na ginagawa mo na wala pa naka-inoculate na organism, you can put it one in the holding area. Or pwede din lagay mo dun mga plate na na-inoculate na, tapos after incubation with your chamber, for example, okay, then it's waiting for you then to observe the morphology. So you can put that one in your holding jar para hindi siya, again, ma-expose sa oxygen natin. Okay, so pag ang plate natin, for example, ay meron na siya naka-strict na organism natin, it should stay only here for not more than 1 hour with our holding area or holding jars. Okay, so you need to put that one in your chamber or eventually you can have it evaluate the morphology. Because more than that, baka ma-contaminate na siya ng oxygen natin and they will die again in the presence of the oxygen. Again, regardless if that one is your chamber, if that one is your jar or bags or even your holding jars, so most likely still ganito pa din ang content niya. Okay, all but one help you to provide an anaerobic environment that is free from the oxygen.